Hello, everyone. Welcome to our Public Health Plus series. We are back with Dr. Mina today. We're going to talk a lot about the intersection of public health and clinical medicine. As Diego mentioned, my name is Michelle James, and I'm the Senior Director of Alumni Engagement for the Rollins School of Public Health here at Emory University in Atlanta, Georgia. Our Public Health Plus series was created by the Rollins School of Public Health Alumni Board's Career Development Committee in partnership with our good friends at Orange Sparkleball to really examine the spaces where public health intersects with a variety of other disciplines. Our goal through the series is to highlight those working at these intersections, discuss their work, and hopefully broaden our thinking. Today, as I mentioned, we're exploring the intersection of public health and clinical medicine. Public health and clinical medicine are often thought of as similar, and some people even confuse them, but they have slightly different objectives. Public health is more population level thinking, whereas clinical medicine really focuses more on the individual health and well-being. So what happens when those two objectives are at cross purposes? So today we're going to talk about, especially during times in a global pandemic like COVID-19, how we can use things like rapid tests as public health tools aimed at stopping transmission, rather than the diagnostic tool of individual medical care. Facilitating our discussion today will be Meaghan Kennedy. Meaghan earned her MPH in epidemiology from the Rollins School of Public Health. And after conducting HIV research at the CDC for several years, she discovered the world of design thinking. So blending her love of public health and innovation, she founded Orange Sparkleball, an innovation and impact accelerator focused on moving public and private organizations' initiatives forward. Meaghan is a current member of our Rollins School of Public Health Alumni Board. She is a mentor for social entrepreneurs and is frequently invited to sit on innovation and entrepreneurial panels. Our special guest today is Dr. Mina, Chief Science Officer at eMed, a digital point of care platform that enables at-home diagnostic testing for COVID-19. As an epidemiologist, immunologist, and physician, Dr. Mina was previously an assistant professor of epidemiology and immunology and infectious diseases at the Harvard T.H. Chan School of Public Health, assistant professor of pathology at Brigham and Women's Hospital and the Harvard Medical School. We are thrilled to have you here today. Thank you so much for taking your time. I'm going to turn it over to the two of you.
Thank you, Michelle. I appreciate that intro. Dr. Mina, I'm super excited to be here to talk to you. I have been following your conversations mostly on Twitter for the last couple of years, and I'm really excited to kind of dive into this public health clinical medicine intersection. But before we do that, Michelle gave quite an overview of your positions, but we typically like to start with a little bit about your background and kind of your journey to where you are. So can you just give us a little bit of that?
Sure. Well, it's I don't have the most straightforward journey to where I am, I suppose. But in brief, I have an engineering background, and that was my initial schooling at Dartmouth was in engineering, and I ended up leaving Dartmouth and moved to Sri Lanka, where I became a Buddhist monk in 2004, and that was also the year that the Indian Ocean tsunami hit the country, and so I ended up leaving being a monk, where I was up in the mountains, and moved to a refugee camp from the tsunami. Lived there for about half a year or so. And that I think is one of the major, I bring it up because it's one of the major pieces of my life that kind of put me on a path of converging my engineering kind of interests with public health and medicine. And so I ended up going to Emory for an MD and a PhD. I spent most of my time in my PhD not at Emory, but initially at the CDC, and then in South Africa, and then at St. Jude Children's Hospital, and then back at the vaccine center at Emory, working on influenza and bacterial co-infections and live attenuated vaccines. And then ultimately, I finished medical school. I did postdoc at Princeton in evolutionary biology and ecology of infectious diseases, and worked on measles and discovered something called measles immunological amnesia. And that's where measles infections kind of destroy a child's or anyone's immunological memory. And then I came up to Harvard to finish, to sort of round out that work on measles immune amnesia, which initially at Princeton was really based on mathematical models and population level data. And then I moved into sort of biotech sphere to develop the immunological tools needed to really be able to prove out that was happening within the individual. And so ran some clinical studies and developed a lot of biotech, and did my training in pathology at the Brigham and Women's Hospital at Harvard. Ultimately became a professor at Harvard School of Public Health in epidemiology and in immunology and infectious diseases, and also an assistant professor at the medical school in pathology, where I started that just before the pandemic started, really. And pandemic kind of was another curveball for the entire globe. And ultimately, just a few months ago, I decided to change course again and become the chief science officer at eMed to really start implementing a lot of the research that I was doing throughout the pandemic, and not just research, but really advocacy and advising throughout the pandemic that I've done for governments and other entities throughout the world.
Thank you for that. That was even more of a curveball than I anticipated in terms of path. And just so you know, typically we have people as guests who I would consider, from my background, I would consider innovators. And they almost always have a story where it's, I started here and then I went here and I went here. So that was my anticipation about how you were going to say that. So it does not surprise me that you had that well-rounded background, I'll say.
Well.
So can you, first, before we start, do you consider yourself a champion of public health? Is that kind of where you see your identity?
Well, I certainly have always really pushed for public health, and have always focused more heavily on public health over medicine in many ways. When I was in medical school, I got frustrated with sort of medicine at the individual level, and I started doing a lot of work, started a nonprofit in Nicaragua to help individuals and communities in Nicaragua to learn how to convert table salt into chlorine so that they could purify their water using solar power. To do the conversion from salt to chlorine. And that was really focused on, I was kind of taking some experiences in Nicaragua that focused on medicine and getting a bit frustrated with sort of the one person at a time approach there, and converting that using engineering principles into public health action. And so yes, I would say that my interest, maybe more than anything, I would say I'm a champion of science. I think science is what drives me, and sort of learning and discovering new things is the most exciting aspect of life for me. But converting that into public health is really what I think is the most important component of all that.
I love that answer, because it sort of sets up something. And when we did our prep, you did say that it was okay if we freestyled a little bit. So please tell me if
Sure.
I am freestyling too much. But I've heard you mention a really broad range of things. So engineering, public health, thinking about the individual level, kind of clinical medicine. One of the things that we talk a lot about is this ability to sort of look broadly and integrate or synthesize a lot of pieces to solve problems. And that's a rare skill set. Is that something that you've always been able to do?
I think so. I think it's what I've always focused on, for sure, is how to bring different elements together in a way that, you know, maybe isn't always being done. To me, it's where exciting new things happen. if you really focus just on sort of what's been happening, the one course, I think it can get a little bit boring. And maybe I've been like really focused on tethering disciplines together, mostly out of just always not necessarily having a focus just on one specific thing, maybe with the exception of when I was a monk, I suppose, meditating. But even there, I think it was really the combination of like the transition from being a monk to the tsunami hitting and moving down to the coast and really kind of brought in a lot of different facets. And I just think that the most important challenges are going to be solved today and in the future by bringing disciplines together. I think we've done a lot of fundamental research over decades and centuries and millennia, really to understand pieces, but the real changes that have happened in society have come, I think, when disciplines are woven together. And it's what I personally just find the most inspiring and interesting, I suppose.
Super interesting. That's something that I think an awful lot about as well. And so I'm inspired to hear you talk like that. So thank you for letting me freestyle. Okay, I promise I will get to the topic at hand.
That's whatever you want.
It's very sweet of you. Thinking about the connections between public health and clinical medicine, can you talk a little bit about where you see healthy connections and where you see tension as a lead into kind of getting into the testing arena?
Sure. I mean, ideally, good public health creates good medical outcomes, and hopefully keeps the need for medicine out of the equation. With the exception, I would say, is preventative healthcare medicine. Not really. It's preventative healthcare. It's it's a component of public health and medicine. And I think medicine is sort of what happens when public health fails sometimes. But one of the big tensions that we see, especially in the context of infectious disease pandemics like we've been in, is that medicine and public health are not always the same thing, and they're not always even in sync with each other, despite kind of you would generally think that they are. But as a very simple example, without getting into COVID right now, let's take a vaccine. Let's take measles vaccine, which is something I've worked very hard on in the past. If we look at it through the public health lens, it's very obvious that everyone does better if everyone is being vaccinated. From a public health perspective, it's just obvious to keep measles at bay. We need to vaccinate everyone. On the other hand, if you were to take any one person out of society, or in that society where you have very good herd immunity, and you look at them as an individual, and you say, is getting this shot in your arm the best thing for you right now as an individual in a society where, let's just say, every single other person is vaccinated? Well, no, that individual isn't going to themselves benefit really from that measles vaccine. But we give vaccines because we understand that collectively the benefit is absolutely there, and the damage if people stop getting vaccinated is immense if we start letting measles come back. And so for that one individual in a bubble, maybe the best medical, if you will, practice is not to stick that needle in their arm if their chances of getting measles are pretty much zero. That assumes that they never leave, a vaccinated community and all that stuff. And so we know that there are these tensions, and there are certain tensions that we don't even think about anymore because it's just so obvious that measles, ultimately, by everyone getting vaccinated, it ultimately does pay dividends from a medical and a public health perspective in the future. So we've come to that conclusion that that's the best medical and public health practice. But that is one area where in isolation, that's a tension. Another area, which does get towards COVID, that has been, I think, a real confusing point for people is, what is the purpose of testing? And is it a medical test? Is it a public health test? And without trying to delve too much into that question yet, because I think you have specific questions you want to discuss, just the notion of isolating somebody, of having to go into isolation. We ask people who are positive for COVID to isolate, but that is absolutely not a medical practice. that is purely a public health practice. At least I've never met a doctor who has prescribed isolation for somebody for their own benefit. It doesn't give you any benefit to sit in a room and isolate away from everyone. It gives your family a benefit, and it gives the people around you a benefit so you don't infect them. So that's actually an area where there's a real tension. But it's again almost so obvious that we don't question it. We just say, of course you should isolate, and doctors recommend to their positive patients to isolate. But actually, that's a public health action that's antithetical to medicine in many ways, because it's not on the doctor to, from a medical perspective, if your focus is just that one person, then probably isolation is not really, leads to depression, it leads to all kinds of anxiety and other issues that are not really in that person's best benefit. So I think starting the discussion, usually we just kind of assume that these things are understood and well-defined. But the pandemic, if it's brought out something for me personally, is that the distinction between medicine and public health are exceedingly poorly defined. And I think it's actually led to some really bad decisions and a lot of excess death throughout this pandemic by not clearly defining what space we're living in, especially when you then consider politics and regulation in the scope of things.
I think you foreshadowed very nicely the kind of diving into the testing specifics. I do want to kind of put a bookmark in the what do we do about that? And how do we define public health more effectively? My bias, I'll say, is I think we need to define public health more effectively, because I think clinical medicine is much easier to think about. It's easy to think about, what do we do for this person to make them well?
Right.
We'll come back to that. Okay. So over the last 2 years, the reason that I became aware of you, and I teased you earlier about this, but was because you were very active on Twitter talking about rapid testing really early on. And for those of us who are epi Twitter geeks on Twitter, it was a pretty active conversation. You're going to do a way better job than I am at describing the difference between a PCR test and a rapid test and sort of the testing landscape. But can you just set that up to make sure that everyone's with us? And then I want to go into kind of that. I called it a battle when we prepped, the battle you've been fighting for the last 2 years. And I do think that's accurate around the use of rapid tests.
Well, sure. So the major difference between an antigen test or a rapid test and a PCR. So a PCR test is a very high sensitivity test that's generally performed in a laboratory, though now we're seeing some PCR-like tests, which can now potentially be used at home. But they amplify segments of DNA or RNA. And so we use them in forensics and in sequencing all the time in medicine for all different purposes. But when it comes to COVID, it's looking at PCR as amplifying a fragment or a piece of the RNA. It's using little probes and primers that actually go and find little, very, very specific sections of the virus's RNA, and it amplifies it. And the amount of amplification that's required to occur before the machine detects that there's actually little particles being amplified is actually reflective of the amount of virus that's there to start with. So if you have a lot of virus to start with, it has to go through fewer and fewer cycles of amplification to give you a readout that says, yes, there is COVID here, or there's SARS-CoV-2. An antigen test, on the other hand, is not looking for the RNA or the DNA. It's actually looking for a protein. So just like we have DNA, but then we also have cells and proteins in our bodies. An antigen test is actually looking for a physical structure of the virus. And in general, they're looking for a part of the virus called the nucleocapsid. And that's just, it's just a antigen test could be targeted against many different parts of the virus. That's just an abundant protein. And so it's a good target because you want a more abundant protein, so it gives you more chances to detect it. And there's no amplification, which means that it's necessarily only going to turn positive by a visual inspection of a paper strip So that you have these rapid antigen tests and they flow along. And if there's virus there that gets grabbed by it's actually antibodies on the test that are printed there by the manufacturer. They're like little antibodies that bind the virus. And then if they bind it, they actually immobilize the virus, and then there's like a second, you could think of it like a dye that comes in and causes the line to turn dark. And the darkness of that line is also a semi-quantitative measure in a way. It actually tells you how much, if it's really dark, there's a lot of virus. If it's not very dark, there's not much virus. But nevertheless, it's because it is lacking in an amplification step. Like, it doesn't, if you think about RNA or PCR, it's almost like a zoom button on a computer versus not having that zoom button. So you're kind of stuck with what you got. It means that it will only turn positive if there is really a lot of virus there. And in general, that's why I like to think of an antigen test as a test that is going to turn positive when somebody is actually at risk of spreading the virus. You can almost think of it like a contagiousness indicating test versus a PCR test might be positive when you're contagious, but it also might be positive for weeks after you've been contagious, just like a forensics detective can use PCR and go in after the crime has been done. They go into a crime scene and they pick up a piece of hair and use a PCR to amplify the DNA in that piece of hair. So it can be used during the fact, but it's also slow. And so it's actually not, if we put it in the context of a crime scene or a crime going on, PCR is a little too slow to actually, it's better almost after the fact rather than during it. You're not gonna have a forensics detective go in and try to break up the crime as it's ongoing. That's more for the security guard to do. So they have two different uses. A rapid antigen test is like for immediate use, and its limitation is that it's not quite as sensitive. And a PCR can kind of be used for immediate use, but really it's slow and it's more methodical and it has to go out to a laboratory, but it's really sensitive. And so that means that the qualities that they each bring to the table are quite distinct.
I love that analogy. The rapid test as a security guard is so perfect. Awesome.
Yeah, I like to think of it, I try very hard to put as much in the biological realm into things that we can envision, I suppose.
Totally. Yeah, no, that was great. I just wrote it down. Okay, so thank you for that. So basically the way that I thought about it as I was initially seeing you championing rapid tests was this would be a great tool so that if I went to, for instance, go visit my elderly parents, I could figure out whether I was infectious before I went in. It seemed very simple to me, so that the chain of transmission is broken, because obviously I wouldn't go if my second pink line showed up. I have been very surprised over the last 2 years what a battle that has been. And I really, in my mind, you're sort of the face of that battle. I know that you're not the only one who's been pushing rapid tests. And I know in, for instance, the UK, they've been widely used. Can you talk a little bit about, and you can go kind of as personal as you want, but what that has felt like to be kind of, it looked like thrust into this position of being the face of something, and then being kind of beat up a little about it was what it looked like from my perspective. I hope that wasn't too much.
No, I think it's, I think you're absolutely right. It has certainly felt at different times like a battle, especially at the beginning. And even the UK was resistant. I was advising the Prime Minister's team in the UK way back at the beginning, and they were very receptive. So although there was resistance, there was also a receptivity to change and to think more broadly about public health than we necessarily saw here in the United States. And that's really to get to your question before about the distinction between public health and medicine. The greatest resistance really came from physicians as far as rapid tests are concerned. And actually it started, so my sort of role in testing started before. I helped get, I was a medical director at the Brigham in molecular virology, and so I helped get the Brigham's testing started. And that was actually a challenge to get PCR testing started at the Brigham. So at that point, the hospital leadership was not recognizing that there was going to be a need for COVID testing. This was back in mid-January or so when I really started getting the Brigham to have some willingness to start a testing program. And I just couldn't, it was really then, I would say in that experience that got me on this sort of 2+ year crusade around testing, because that was me wearing my epidemiology hat. And so my colleagues at the Harvard School of Public Health, the people in my laboratory, there was sort of a pretty large group effort to understand what was happening in China. And it became very clear once the virus was in every province of China, within a few weeks, that it was going to be a pandemic. And it was amazing to me that the hospital leadership was so unwilling to bring a COVID test on board and give me the resources to build it there. And it was really, that started the battle. And then I started working with the Broad, realizing that we were going to need even more testing than just the hospitals could provide. There's an institute called the Broad Institute, which is part of MIT and Harvard, and it's essentially an engineering meets biology institute, if you will, with a very high efficiency biology group that usually does genetics, human genetics. And so working with them, I said, can we actually use this facility to do COVID testing? Because we're really going to need a lot for just Boston alone. So that grew into one of the highest throughput, sort of largest PCR labs in the country, and one of the most efficient. But it was really that made me realize, hey, even this is not going to be enough for a pandemic. We have to figure out some way to get faster turnaround times. If you have a 3-day turnaround time for a PCR and your goal is to stop somebody from transmitting, then 3 days is not sufficient. It needs to be 3 minutes or 30 minutes. And so really with some folks in my laboratory, we started looking at the science of how the virus grows inside of people based on what we knew of other viruses, which is really sort of my background, and recognized pretty early that the virus grows across many orders of magnitude. So you don't need super high sensitivity testing like PCR if your goal is to stop transmission. And if your goal is to stop transmission, you actually just need to have pretty mediocre sensitivity as long as the test is fast, because then you actually get actionable results in time to make a difference in that person's transmissibility. And ideally, it starts turning the test positive when you need it to, which is when people start to become infectious. So we actually, we did quite a bit of research, both on how to better sort of implement PCR testing strategies and how to implement rapid antigen testing early on. But all of it came down to optimizing on something that is not normally discussed by medical doctors, which is transmission. And transmission is not something doctors are trained to really think, or, I hate to say it, but care about, because a doctor's view is I need to treat the person in front of me. I'm not treating the people I don't ever see them interact with on the bus. That's not my job as a doctor. That's my job as a public health person. And so optimizing a testing strategy around a public health metric is actually kind of a new thing. And doctors, I think, unfortunately, took a very long time to adjust to that. Because a doctor, I put it in the context of TSA agents at an airport. And this is not to suggest that there's any likeness between the different professions, but a TSA agent at an airport who's watching the bag go through, their job is to make sure that that bag goes through the X-ray machine and is getting the best view of it possible, but that their job is not to ensure that there are one-way doors and security guards and the whole security system of the whole airport. Their job is just to deal with that bag as it goes through and then move on to the next. And it's sort of similar to physicians. A physician's job is to deal with the patient in front of them, not to think broadly about the whole ecosystem of testing and the system of reducing transmission at a population level. That's not the kind of training that a physician gets in medical school, nor residency. But a lot of the decisions were because doctors are generally the ones who are in charge of testing in a hospital, for example, they're the ones who were being asked all the questions about what tests are best and what to use for a pandemic. And so it led to this massive tension. And there were very few of us who had both the expertise in medical testing and we're also epidemiologists and sort of thinking more at a mathematical population level. And I think that's what set me up to kind of be you could either say I was the face of this battle, or you could say I was like really, blacklisted from the pathology community in some ways for quite a while, because I was advocating for something that was seen as inferior from a medical perspective.
I would also say, so you talked about the medical profession, and well, I'll go ahead and say it, not being kind of trained in what I would call systems problems. And then you kind of leaned on your epidemiology training. I didn't see a whole bunch of epidemiologists jumping on board with the idea of rapid tests. So my background is epidemiology, so I'll raise my hand and saying that I identify as, partly as an epidemiologist for sure. I was pretty disappointed in a whole bunch of epidemiology folks because I didn't see them understanding. They wanted to discuss sensitivity a whole lot. Like they relied on their epidemiology training. So what is that component that allows you to look at something from a systems perspective? Decide, yes, I have this training, I have clinical medicine training, I'm going to take what I need from that. I have epi training, I'm going to take what I need for that. I'm going to synthesize it here and be able to make the best decision for this moment. I think that's a different skill set, and I don't think you've hit on it yet. Do you think about that?
I think about it a lot. I don't know what, I was never trained in it. I think in some ways I did receive the training that I needed to do it, in that I had a very sort of like I said, my undergrad was engineering and public health. My PhD was a mix of mathematical modeling and creating live attenuated vaccines and bacteriology and clinical studies. And then my PhDs were mixed between math modeling and epidemiology, and then developing phage display technologies to display epitopes for immunological technologies. So, I think it's really diving deep into each of these different topics it starts to make certain things obvious that might not always be obvious to somebody else. And one of the areas that this really became apparent, like you said, there weren't a lot of epidemiologists working in who were really getting on the train of saying, hey, we should be really advocating for rapid antigen tests because they can be scaled up and because we don't have vaccines yet and things like that. And there were times when I was practically begging epidemiologists in my orbit to back me on this. And in general, what I found was there was a too little, people didn't feel comfortable talking about testing because it wasn't their expertise, which is great. I really feel pretty strongly that people should not, be pretending to be experts out of their area of expertise. But what it showed me is that while epidemiologists generally work with diagnostic test results, they get case numbers and things like that, there isn't a good education in the biology of a test. So when I'm sitting there saying, no, we don't need PCR, we need this other kind of test, most epidemiologists who have never really, most epidemiologists don't do laboratory diagnostics in the healthcare space, and a lot of them have never actually really spent a lot of time pipetting or, working with physical molecular tools that diagnose things. And I think there was just a discomfort to wade into the conversation because they're hearing people that they have always, probably admired or felt a genuine respect for, who are pathologists in the medical space, saying, no, PCR is the way to go. We can't use other things. And then they have me and a few people saying, no, PCR is great for certain things, but we need this other technology that everyone else is saying is inferior. And I think there was just this discomfort by epidemiologists who didn't focus on this technology space to wade in at all. And I think that's a different issue. I think what I saw in this pandemic is I don't think we train epidemiologists. I don't think we train anyone in a sufficient way in the graduate training to really to really focus on the cross-disciplinary nature that's required during a pandemic. There are some programs that are starting. Emory, I think, is actually quite good at it, at really training people across disciplines and giving graduate students the space. Other institutions really are not. They still live in very siloed departments and siloed laboratories. And for me personally, I think I spent a lot of time very actively crossing scales, crossing bridges, crossing departments in whole different, from mathematics to phage. And but without that kind of pretty in-depth training in all those fields, it would be very hard to feel confident enough to go out on a limb and say, pathologists are wrong in this case. We need a different kind of test. That was like, it oftentimes felt like me against the world, or me and a handful of people against the world.
I really appreciate kind of you talking through that. Again, we are freestyling here a little bit, so push back anytime. I have made another note as you were talking. When you talked about consulting with the UK, you talked about receptivity to change that you didn't see necessarily in the US. Innovation is what I do. I have a lot of thoughts about all of this. So I'm kind of leading you a little bit here, but so you talked about having broad training, and that helps you kind of pull in the synthesize across. What is that receptivity to change piece? Have you thought about that?
Yeah, I think you almost need, I don't know if we need training in change, but certainly if we go back to elementary education, K through 12, and college, and in many ways, and medical school for sure, we train people to believe that what's in the textbook is right. We don't train people to think. And so in ninth grade, I got frustrated with the public school system, for example, and I decided that I wanted to go to a Waldorf school. And a Waldorf school is sort of a totally different style of schooling. And it teaches you, instead of teaching you physics equations, you learn first all about who the scientists were that came up with those equations. And you start learning things from the ground up. And I think that stuck with me, that has always stuck with me, to ask questions and not take things at face value, but to derive them yourself, make sure you really have a deep understanding of them before you go and espouse them to others, if you will. And but our whole educational system is focused on a very different type of thinking for the most part, which is memorizing, regurgitating, and kind of spitting out facts that you have heard. And that makes it very, very difficult for two reasons. I think on the one hand, you are trained to believe that what you have learned is correct, which, is an interesting point unto itself. But you're also not trained to really think about how to get yourself into a different headspace to think about the same thing in a different way than you've always thought about it. We don't have that explicit training of change. And I would say a PhD type of training does go in that direction quite a lot. You're trained to deal with adversity in the problems you're dealing with, and you're discovering things. And ideally, if you have a good PhD experience, you run into things that you had a preconceived notion about that turned out to be the opposite or false, and you have to, but that's not how doctors are taught. Doctors are taught in a very different way, which is this is right, this is wrong, or this is the biology, this is the study. And it's really hard to change medical dogma. It's very, very difficult. And in some ways, it's the people who go into medicine today, are selected by tests. And we select medical students from undergrads, we do MCATs, and we do all this memorizing type of experience to get people from point A to B to C to D in medicine. And that sets the stage for what I think is it ultimately selects for a type of brain that might be really good in one aspect, like memorization. Me personally, I am not good at memorizing. I'm really bad at it, which is probably why I gravitated towards engineering and mathematics, because you can derive things. And I do think I worry a lot about our medical world. And the way that we train physicians and really the way that we select who will be the next generation of physicians is so focused on your ability to memorize today that I do worry about how good of problem solvers will we have in the future as our physicians. And I think the pandemic, to me, demonstrated that we have a lot of people who are very good at problem solving in one specific way, but maybe aren't able to translate their knowledge here over to something a little different. And I don't think we train that in people.
It's to your point about it, medical training is about it's this or this. One of the first kind of threads I was reading was this discussion of PCR is the gold standard, so we don't need to talk about anything else. And it was very much, yes, of course, it's the gold standard, but for what use case? And so that was when I originally kind of got on well, this is going to not go well.
There's a good example. I mean, still our regulatory landscape has not changed at all. For the most part, we still have high sensitivity PCR as the gold standard, even if it takes a week to return a result. So we would prefer from a medical approach, we would prefer during this pandemic to have, and I wouldn't say we did prefer, in the United States to have a whole nation of tests that took 5 to 10 days to return, and ultimately were effectively useless for public health purposes of transmission mitigation over an 85 or 90% sensitive rapid test. So we could either have a rapid test that's 85% effective, if you will, or a PCR test that's 0% effective, but will ultimately give you the right medical answer eventually. And we chose the 0% effective route. And we are, to this day, that's still kind of the gold standard for what? That's the gold standard for isolating people when they are no longer infectious. Great, go with PCR. But we don't want, in public health, it's the wrong thing to do to tell someone they're positive and need to isolate when they are no longer infectious. But we have a lot of data actually that shows that in Massachusetts, for example, through the summer, let's say June and July of 2020, this is just the data that I'm thinking of at the moment, the vast majority of people who were told to isolate were told after they were infectious, when they no longer needed to isolate. That's bad public health. So while it might be a great medical diagnostic tool for a physician to say, yes, your symptoms, your stomach problems, your respiratory problems last week were probably from COVID because you have remnants of virus RNA in you now. From a public health perspective, it's the wrong thing to do to ask that person to isolate when they are no longer a risk to their neighbors.
For sure. I am sensitive about time. I have 1,000 other things I could ask you, but I do want to, if other people want to put questions in the chat, that's totally fine. I will pay attention to that. And then, Michael, if I can just talk to you a little bit now about what you're doing now. So you jump from academia to industry. I'm going to foreshadow what I see. I don't know if you see it this way, is a continued broadening and maybe a knowing now that you have an engineering background, which I didn't know before, kind of re-embracing some of that, but I don't know if that's the right interpretation of kind of your latest move, but could you talk a little bit about that?
Sure. Yeah, it's. I think it is very related to my engineering background. I've always wanted to build things, building sustained public health initiatives, whether that was in Nicaragua with the solar panel to chlorine, saltwater conversion to chlorine, and we were creating little micro businesses or teaching people how to create these micro businesses for themselves. And it was something more sustained. And I view what I found as frustrating in academia is you can't really build things easily because there's a lot of reasons, but some of it is you have a fairly transient workforce, meaning you have PhD students and postdocs who cycle through. And each of those individuals have very specific skill sets, and they to move on to their next new phase, they have to be the first author of their paper. And there needs to be a last author, and it's very difficult to really build a system when you have each person needing to own their own little piece and go off and have it be novel. Everyone, because that's academia, everything has to be novel and a first and has to lead to a research paper. And a lot of the reason why I got into advocacy around rapid tests was I felt, we could write papers, and we did. We were very successful. The postdocs and PhD students that I had in the lab are amazing, and we're very, very good at doing research. And we put out some really nice research papers throughout the pandemic. And some of those are slowly changing the way we deal with the public health aspects of testing and other aspects of this pandemic. But ultimately, I found that my advocacy work was where I was feeling like I actually was reaching people who were not academics, or when I was advising the White House or advising the Congress and Senate and such, I felt like that was more action oriented because it could actually lead to something. And building something has always been very, very important to me. At Harvard, I was thinking about building what I call the Global Immune Observatory, a network of laboratories around the world that would be able to really enhance surveillance. I've been working on that since 2015 or so, but I found it was very difficult to do that, to really build that kind of program within the constraints of academia, which are really focused where the currency is a paper and not a product that helps people. And the expectation is that your papers go on to help people secondarily. And so what I found here coming to eMed is, the next transition in this pandemic for testing, now that we have everyone up to the President of the United States talking about rapid tests, I feel like at the end of the day, I feel like the advocacy has been a success, if not an exceedingly challenging one. It's now, what do we do with those tests? How do we actually enable people to really benefit from them? Because we're entering this new phase of saying, okay, a test can be used for public health purposes. You use a test before you go to visit your family so you don't get them sick. But wearing my medical hat, people also do test for medical purposes for their own benefit. And now we have oral antiviral therapies. And the question is, how do we make oral antiviral therapies and future medicines accessible to people without the major barriers that we've created in the medical industrial complex of the United States? It's scary to go to the doctor because you don't know if you're going to get a $50 bill, a $10 bill, or a $5,000 bill. And most people don't have PCPs, primary care physicians, anymore. They go to the emergency room, and there are massive, massive barriers to entry to medicine. Can we take all of this effort that's gone into getting tests into people's homes and now translate it into enabling that to become a process where somebody actually has access on their own terms to a test that can actually be then translated into treatment to make them or their kid better? So I think of, for example, a mother or a parent and their kid with strep throat. Right now, it's a major burden on people to have to say, my child has strep throat. I have to bring them into the pediatrics office. I've spent half a day or a full day off of work to do that, get the kid tested to find out that they have strep throat, which you probably already knew, and get them a prescription and then go back to home. What if we could do that whole process by giving somebody access that they just pick it up at CVS, or they already have it in their cupboard? They have a strep throat test for their kid. When their kid has strep throat, they pull it out and they use it on their kid, and it can immediately, if they're positive, lead to a prescription for amoxicillin. Something as simple as that we can use technology in the 21st century to actually really start reducing the burden of the healthcare costs in Americans, I think is not just about cost, but it's about access, and it's about speedier access to treatment and clinical studies and trials. And so that's kind of what I'm working on here at eMed, is to give people access to things that are currently not really that accessible. And in COVID, it was, you had a choice in 2020. Do you go stand in the cold in a line for 4 hours waiting to get a PCR test, or do you not get tested? And now we're in a very different world where you can have a test in your cupboard for when you need it. And that's what I'm trying to do here, and really take this idea of test to treat. where it's one seamless initiative where you can wake up, you have a scratchy throat, use a COVID test. If you're positive, there's a laboratory report that immediately gets provided to a telemedicine and you get a prescription in a half hour. If we could do that, then we've greatly, greatly reduced the barrier to entry for somebody to not land themselves in the hospital.
I think you have a convert here. Your use case is exactly what one of our colleagues is dealing with. So basically what I'm hearing you say is now you've left academia to, and I'm going to use startup language here, but to disrupt clinical medicine.
Yeah, not necessarily disrupt clinical medicine as medicine, but to ease to break down barriers to keeping people healthy. And I think if COVID has taught us anything, I think if there's any human right that we should have, it should be knowing what's going on inside of our own bodies on our own terms. And should we have to go to a doctor every time we want to know about what's happening in our body? Some things, yes, we should, we need a doctor to explain it, but some things, no. Knowing if you have COVID, for a full year and a half in this pandemic, every single COVID test that was given out had a prescription associated with it. There was some doctor behind the scenes getting paid for that test. You never talked to the doctor. It was just a waste. And and people had to go out and get that test done somewhere. And I just think it's people's right to have access to knowing what's happening. So I don't think it's not necessarily totally disrupting clinical medicine, but I think it's the process by which people enter into the clinical medicine world needs disruption.
Delivery, for sure.
Yeah.
Thank you. Michelle has a great question, and it seems like we're getting close to time, but I'm going to dive into Michelle's question. And I think this is a great question, Michelle. How does CDC factor into this conversation? So historically, CDC has been the place where clinical and public health expertise are synthesized, and guidance comes out of that. And we might not have seen kind of that working in the way that we expected it to work during the pandemic. Do you have thoughts about that?
I sure do. Yeah, I think what we've seen is a reflection of the erosion of public health and public health thinking and medicine, and being able to bring them together in a really constructive way. We have seen massive blunders. We've seen data being used, medical data being used to drive public health policy, which, as we've talked about, is not always the right thing, especially with testing, for example. We've seen public health data being used to drive individual medical policy. And I think what we are seeing is a pandemic is not it's not a time to be tunnel visioned. It's not a time to keep any silos up. It needs people being able to talk across the table to each other. It needs economists and public health people and physicians to all carry almost equal weight. And what we've seen is a real lopsided weight placed on different areas of expertise in the CDC during this. And I think it's a reflection of sort of how we've recruited people into the CDC for many, many years. And it's also a reflection of public health in general being eroded and not putting the right focuses in the right place. Like I'm always, I always ask the question, is it the most appropriate to always have a physician in charge of the CDC? Should there be two people? Because a doctor's education is not, again, it's just generally not about optimizing on public health metrics, optimizing on mass metrics. It's optimizing on individual metrics. And those can sometimes, unfortunately, they sometimes do conflict with each other. And so I think that what happened with the CDC in particular is that we have not well defined what our goals have been. And that what that meant is we were continuing to this day to ask the right questions to the wrong people or ask the wrong questions to the right people, and we have rarely had the right questions be asked to the right people. And I think everyone, like a lot of people who have chimed in during this pandemic have had the right answers, but to questions that maybe were being asked at a different time of the pandemic or something. And I think that has been, we haven't had clarity in terms of what we're talking about. Something as simple as, are we talking about somebody's medical, one person optimizing on one person's medical well-being? Because yes, we can put all the resources in the world into making sure that this one person gets the best medical care they want. But what happens in public health, and what happened with the FDA and the CDC, is inaction is rarely given the same weight as action. And what I mean by that is, let's say in the middle of a pandemic, when we have people dying on a daily basis, we have to weigh what are the consequences of inaction more heavily than what are the consequences of action that carries a little bit of risk. If the large majority of people will be benefiting from X, Y, and Z, then maybe that risk is okay if it means that they're all getting treated or vaccines or something, when the alternative is do nothing. And that is what we've seen with this like inability to weigh inaction, because inaction is a natural process. It's like, well, you can always blame that on nature. That's the virus. Nobody gets blamed when, rarely gets directly blamed when people are dying from inaction. They get blamed when somebody dies from an adverse effect of an action. And I think that, we don't have a good way to balance that in society.
I think that's it reminds me of not having looked at the history of pandemics. And I could have taken that soundbite and talked about HIV exactly. And it seems like we just don't learn the lesson. It happens again and again. On that note, a very optimistic moment. We are at time. There's one more question. I will leave it to you whether you have time to, oh dear, now maybe there are two. I want to be very respectful of your time, so I'm not going to push you on questions.
Well, I can answer these if you would like. I'm happy to right now.
Okay, thank you. There's no pressure at all.
No, I don't feel any pressure. I will.
Okay.
So absolutely, to Philip's question, a bias in in studies measuring the sensitivity of antigen tests against PCR. I'm pretty sure that we were probably the first group to really talk about the real problems there in that bias and what it is we're trying to measure and what it is we are like. Are you actually? Do you have a false negative when you have an antigen test that does not turn positive, even though a PCR test is turning positive if it's 2 weeks after somebody's been infectious? I would call that a false positive on the PCR and not a false negative on the antigen if our goal is identifying people who are infectious, because that person is no longer infectious. And so absolutely having a single standard for the gold standard, regardless of use case and regardless of why we are testing, has been one of the most damaging things we could have done. That's why earlier in the pandemic, Christian Anderson and I wrote a paper in Science that to us is just super simple, but it's called for COVID-19 testing, one size does not fit all. And it was just to discuss this issue that we have to be careful about saying that something is the gold standard test, because sometimes too much sensitivity ultimately ends up creating false positives, especially in case they're coming down in the community, and all kinds of issues, not even to mention the fact that it really inhibited antigen tests in this country from becoming available, because we just assume that a positive is a positive is a positive on PCR. And if it doesn't live up to that, then it's failing. And that's just an absolutely wrong view. It's a medical view, because a doctor is more like a forensics detective. A doctor wants to know when their patient comes to them that you're assuming that you're testing for a reason. So of course you always want the highest sensitivity test, because you want to be able to tell that patient, yes, this is probably COVID. But if your goal is to say, if you're doing the test because you want to tell them whether they need to isolate or not to keep those other people around them safe, then you want the security guard there. You don't want the forensics detective. And these are two very different issues. There was another question about with the shorter incubation of Omicron, how often do rapid tests need to be given? So it's not so much the frequency. So the most informative test that you can do before visiting somebody is the test done right before you visit them. And so it's not so much frequency of testing as more what's the sensitivity of the testing regime. We published in the New England Journal talking about this idea of it's not the sensitivity of the test itself, it's the sensitivity of the testing program to catch the most number of infectious people in the population. But if you have a particular event that you want to go to, or a particular time when you are visiting somebody, the best time to test yourself before doing that is as soon before that event as possible. That's going to be the most informative. And with Omicron, what I would say is there's a very interesting phenomena that's happened, which I started talking about kind of again before we had data, using more of a systems thinking approach, but ultimately we now have data for it. And that was, there were a lot of people saying, hey, I'm symptomatic, but I do not yet have a positive test, and this is strange. And we can now, what happens is when you have a breakthrough infection with Omicron is really good at causing breakthrough infections. So a lot of people, I would say the majority of people in the United States who got Omicron this year, in the past few months, already had some pre-existing immunity to it because of vaccines or because of previous infections. And when you have pre-existing immunity, at the time that you have it, that you get exposed, you're going to create symptoms, but those symptoms are your immune response recognizing the virus, causing a fever, doing the things that it normally does to try to diminish the viral replication. And so it's really important is that sometimes people are now getting symptoms based on the immune response to the virus and not based on the viral damage being done, like loss of smell is viral damage. And so why I'm saying this is that if you feel symptoms, you will sometimes have symptoms before the virus has a chance to grow in your nose. And so now when people are first feeling symptomatic, you might want to wait a day or two before testing yourself if you only have one test at your disposal, for example, because the virus might not actually grow up. You might have only been exposed yesterday. You get symptoms today, but the virus might take 3 or 4 days to actually replicate enough to turn a test positive, including a PCR, which is why if we go back in time, we think during all of 2020 and 2021, we said, do not test yourself for about on average 4 to 5 days after exposure, because it takes a while for the virus to grow, to be detectable by any test. And so now our immune systems are so good at detecting the virus that on day one, we might be symptomatic because our immune system is revving up, but our tests might still be negative. And so we want to make sure that if you feel symptomatic today, assume that you're positive for COVID, even if you're negative, and test again the next day or maybe even 2 days later, but leverage the fact that our immune systems are giving us this red flag saying, hey, warning, warning, you might be infected. We can use that to our advantage, not instead of how it's been largely used, which is almost saying, hey, I'm symptomatic, my COVID test is negative still, the COVID tests fail. We should actually be saying, no, we're actually in a better position now because our immune system is actually warning us about what's happening, and we can use that to our advantage to stop spread.
I love your description of that as I used my systems thinking ability to think about what the answer to that, what was happening before I had the data. So if we could all get to that level of problem solving, I think we'd obviously be in a better spot.
I agree.
Okay, I am not going to keep peppering you with questions. I really appreciate the conversation, and thank you for letting me kind of go a little off what we had talked about and being very gracious about it.
Absolutely. Happy to.